First, I want to ask, okay, recording, okay. First, I want to ask or say, inshallah, you guys are all prepared, and I want to tell you that MSK is fun, and solving questions in MSK, it feels different than other blocks. You like analyze things and, oh, this guy had an injury, so this must be affected. It's a nice experience, but it's not easy, of course. You gotta be prepared and know what you're doing, what's happening. Few ground rules just before we start. There's no really ground rules. I just want to say that if you have any question about the lecture, just stop me at any time. You're not disturbing me. You're not even like stopping my explanation or affecting me in any way. Just stop me and tell me I have a question and say your question, and I'll listen to you. I'll be very happy to help. So don't hesitate to ask. I'm here to help you. So ask whatever you want. So as you know, this is a skeletal muscle physiology, our topic for today. I'll try my best to simplify for you the concept and how things work. Now, I got you slides from Dr. Shams. I believe he's your tutor for physiology this year. So I got his slides, same exact slides, but I will be drawing some stuff to deliver the information to you, and then we'll go over the slides and explain to you some fun things you need to know for the exam, especially. Now, this is the clear slide. I want to draw for you some things, and if you didn't understand anything, stop me and tell me I didn't understand. Do not hesitate, guys. And if you have any questions, stop me and tell me I have a question. Please, don't hesitate. Okay, so I will draw the sarcolemma. You know the sarcolemma, right? Sarcolemma is the membrane of the skeletal muscle cell. It's just called the sarcolemma. For some reason, they gave it this name. I know names are not the best in medicine, but this is called the sarcolemma. And then sarcolemma, we also know it's not a straight line. You know, there's an invagination, or we can say protrusion, or like sarcolemma goes all the way straight and then goes down and then up. Goes straight and down and up. I think you know what I'm going to. There's something here and something here. I don't know why it's doing this. Anyway, now this protrusion of the sarcolemma, it's called the T-tubule or transverse tubule. And these two things around are called the sarcoplasmic reticulum. So just the reticulum and the sarcoplasm. And together they make the, if you know, the triad. You know the triad, right? The triad, okay. This triad is very important. I'll tell you why. Now, you know the action potential comes from the axon and then terminates on the neuroskeletal junction, right? This is the neurons, acetylcholine leaves. Acetylcholine will come here to some muscarinic receptors. You know, you took them, but this is not my topic today. After all of this happens, depolarization comes into the T-tubule, the triad in general, all of them, exactly here. And between these two, there's something really cool about this. Look, there's one receptor here, and I think you know what this receptor is. You should know what this receptor is, and one receptor like this. I don't know why it keeps doing this line. I'm actually annoyed by this line. It keeps going to the left, but anyway. There's these two receptors, they're like this, okay? The receptor, the left one, which is this one, this is the ryanidine receptor, RR. We'll call it RR, okay? Or ryanidine receptor. And this one is the DHT receptor. Now, what happens is when the action potential comes and hits the DHT receptors, the DHT, which we said this is the DHT and this is the ryanidine receptors, will unlock. After this unlock happens, calcium, you know, it's very concentrated inside the sarcoplasmic reticulum. So once there's an opening for it and a confrontational changes, I know this word is very weird, happens in the ryanidine receptor, calcium will leave. And you know what calcium does? Yes, when it leaves, okay, I'm changing this up. 1 second, I hate it. I don't want to use PPT to draw. We said this, and there is action potential here. And calcium leaves, Ca leaves from the sarcoplasmic reticulum, and it goes to the actin and myosin. Here's the actin, here's myosin, You know, there's these myosin stuff that go up. When calcium binds to the actin will expose its binding site to the myosin, and the myosin, look at this. This is the actin, this is the myosin. When the site is exposed, the myosin will grab it and pull it. And what happens with this? The muscle contracts. Now I'll explain to you exactly what happens when and how the troponin is removed, exposing the binding site. 1 second, guys. I'm very sorry, guys. It was a critical. Anyway, we said when the site is exposed to the myosin attached to the actin. Now I'll explain how exactly this happens. Let's go. Now, this is what I explained to you, the triad, SR, and then the transverse tubule, action potential goes there, the receptor is open, calcium leaves, calcium goes to actin, exposes the binding site, myosin binds to actin. There is still some details I will get to you, but this is the general idea. Okay, here's what I said. This is only what I said. And the notes, put them there for you. Actually, Dr. Shams made these, and the notes, they're very good. You can read them to review. This is the action potential. As you can see, let me put the pointer, just 1 second. The pointer, yes. As you can see, it goes here to the T-tubule, action potential, make depolarization, opens the calcium channels, which are the iodine receptor and the DHT. DHT goes away, changes happen, calcium leaves. This is exactly what's happening. And it's all written down in the notes, so you don't have to read the notes now. And this is what I told you, that inside the sarcoplasmic reticulum, it's very concentrated. So once it's open, the calcium just gonna flood out of it. That makes sense. And as I told you guys, if there's anything, just stop me. Here's what I'm talking about, the ryanodine receptor, the THP receptor. After it's depolarized, let me zoom in. After it's depolarized, it will slide, allow the ryanodine receptor to open, calcium will leave. All right, that's cool. That's very cool. And now this, allow me to draw again. I will show you something really cool about this. Let's remove all these. And I want to show you something that's really cool about the process of how the actin exposes its binding site to the myosin. So here's the actin. And you know there are three things, actually 4 proteins attached to the actin. Maybe it's too much, but you'll understand it. I will draw just, this is the actin, okay? And this, what is this? I'm drawing this as a tropomyosin, okay? The green line is the tropomyosin. And what we know about the tropomyosin is that it blocks the binding sites of the actin. So the actin has binding site, tropomyosin, it blocks it. So the myosin can't catch the actin. So this is the blocker. But there are also some things that are holding it in place. The troponin T. See this? Troponin T. T is for tropomyosin. So it's troponin, tropomyosin. Just say troponin T or TNT. It's the same thing. Troponin T, just write T there, is holding the myosin. But there's something that's also holding troponin T. See, this is cool, okay? This is maybe a little bit confusing, but it's actually cool. Look at this. Troponin I. You got it. I'm pretty sure you know it. Troponin I stops troponin T from moving. So it's like, stop, don't move. And troponin T is holding tropomyosin. Okay. What else we got? We got one more thing. Very important thing. Look at this. Look at how cool this is. The troponin C. C for what? Calcium. This is the troponin C. And he's the good guy in this villain story. He's the good guy. He wants contraction to happen while everything is trying to stop contraction from happening and blocking the site. Now what happens? Why is calcium important? See, this is why calcium is important. Calcium comes in from the sarcoplasmic reticulum, okay, after it's opened. When it's opened, calcium will come in, binds to the TNC or troponin C, troponin calcium. Now, when it binds, things happen. They start changing, start moving around, like going crazy, you can say. After they start going around, eventually, what will happen is actin binding sites, myosin binding sites on the actin will be exposed. So the tropomyosin will move away, troponin T, move away, troponin I, move away, allows everything to move away. Troponin C did that. We cool? Everyone with me? Everyone gets the idea? If someone doesn't, please stop me. I know this is too much, so if you stop me, I'll be more than happy to help you. But this is what happened so far. Okay? This is what happened so far.
Can you repeat
And
the first part?
Of course, I will repeat. The first part, which you mean the tropomyosin.
I don't really late, so just like can you like what I missed really fast? Like
Yeah, sure. I will go over everything. Let me just finish this point and I'll go back to you where I started. Of course. Thank you for asking. Now just focus with me here about how proteins move to allow myosin to bind to actin. Now we know that myosin is put red for myosin, all right? Here's one, here's two, here's three, all right? Myosin, as I told you, grabs the actin. Contraction happens. This is what happened here. That's cool. Now, what I said at the beginning, I said that this is the sarcolemma, and this is the invagination, which is a T-tubule, transverse tubule. And then there is the sarcoplasmic reticulums. And between these two, there is the ryanodine receptor and DHT receptor. When depolarization happens, positive, DHT, it's like this. DHT, will move away from ryanodine receptor. Ryanodine receptor would be open now. Calcium will leave ryanodine receptor, and calcium will go here, see, to the actin, and allow the actin to be exposed for the myosin to contract. This is what I said. So far, this is what I said. Okay, we're cool with this. Let's continue from this lecture. Now, here's just telling you about some chains. Also, like the thick filament. I didn't tell you about the myosin chains, but the myosin has like 3 chains. Thick chain, which is basically this one. This is just thick chain. It's the whole thing. And there are 2 small chains. One is the regular to light chain, and one is the alkali light chain. Now the function is written down for you. It's not very significant for you now, but the regulatory chains, they regulate the ATPase. Why is ATP important? I'll come to that in a second, but it regulates the ATPase in the myosin because it hydrolyzes ATP, puts back ATP, and I'll come back to that in a second. And this is what I told you. See, this is what I said. Look at this. See, this is the binding site, and this is tropomyosin. See how it's blocking the binding site? Troponin T is holding it. Troponin I is stopping all of them. Troponin C is trying to do something, but it can't without calcium. Calcium comes in, saves the day, binds troponin C. Troponin C moves everything away from tropomyosin. Tropomyosin will move away with them. Then, boom, you have contraction. There you go. Beautiful. They're beautiful things. So far, everyone's okay? So far, is everyone okay? I'll assume this as a yes, and I'm happy to hear that.
I just wanted to
Okay.
confirm something.
Ah, yes. Yes,
So
say.
like at the end of the day, like the myosin is binding away or moving away, right? From the myosin head, right?
Myosin, this is the head. The head is the one that's binding. The myosin is pushed up. As I told you, this is the myosin. This, my right arm is myosin. This is the actin. Myosin is like this. Wait, let me show you. Myosin is like this. When the area here is exposed, it grabs it and pulls it back. See what happened? Contraction.
Oh, okay, but like are the binding sites then open or
A binding sites of what? The myosin?
like I think that was the actin binding sites
Actin binding sites.
on the myosin, because like he said, and like at the end of the day, what happens when calcium binds? Like after
calcium binds to troponin C on the actin.
yeah.
And when it binds to troponin C on the actin, troponin C will push tropomyosin, which allows, and troponin inhibitor, which allows tropomyosin to move away from the actin filament, which will expose the binding sites to the myosin. Myosin here doesn't have to do really anything. I will tell you what myosin does in a second. The next slide, how it binds exactly.
Oh,
Did
okay.
I answer your question? Did I
Yeah.
answer your question?
Yes.
Okay,
I
that's
got it.
great. That's
Thank you.
great. That's great. Of course, of course. This chart. Now, chart cycles, you know, just no. You don't want to memorize every step. I got you with something really cool. Okay, just stay with me. Focus with me, please. I got you with this one. So normally, this is what's happening. Myosin is relaxing, and the ATP is bound to it. So this is myosin. This is actin. Again, I'm gonna use this all the time. This is myosin. This is actin. Myosin is bound to ATP. Now, when ATP is bound to myosin, you know, it stops the contraction, right? See the last step here, last step, which is step 5. When ATP binds to myosin, it stops the contraction. Now let's start. I want to show you how contraction happens. And it's very important, and I'll tell you why after I just say this step. Myosin is here, and it has ATP. When ATP on the myosin is hydrolyzed, which means it's broken down to ADP plus P, and energy is released, the myosin will go and rotate, get closer to the actin. See, it was here. It was here. This is the myosin. It was here. After ATP is hydrolyzed, it will go up and rotate a little bit. Read it, rotate a little bit to catch the site and put it back to where it was. See, this is really cool. Now, after this happened, this is contraction. Okay, this is contraction. Now, we want to stop contraction. What will we do? We also use ATP. So when we use ATP for this move, which is called the power stroke. Of course, you heard of it. This is the power stroke. You saw this? It's really cool. Hydrolyzed ATP will cause the power stroke. And then when you want to stop the contraction, ATP will come again, bind to myosin, get it back to where it was. So ATP did two things. It caused the power stroke, it got hydrolyzed, myosin, catch actin. Second thing, ATP, when we want to stop contracting, we'll go to myosin, pull it back. Now let's look at the diagram and I show you what I said. See, this is normally, in the normal case. This is relaxed. ATP gets hydrolyzed. It changes the myosin a little bit. It gives it energy. Power stroke. Goes up, boom, binds to actin. Now it's bound. ADP plus P is released, which is phosphate, because it's hydrolyzed. We said that. After I want to stop this, look at this, look at this. Number 5, look how cool this is. ATP comes back, boop, get back to where it was. Very simple, I guess. I hope this made it very clear to you, this cycle. If not, please tell me, I'll just repeat, I'll try to clarify in different ways. And what I said is basically down there that ATP binds, ATP, you know, hydrolyzed. It's written down there for you. I assume everyone got this. Now,
Can you just mention
we
what's
said
power
that.
stroke? Okay?
What's power stroke?
Yeah.
Okay. Power stroke, you know, they're normally like this, the actin myosin. Okay, let me put my hand like this. They're normally like this. Now we need to lift the myosin up to grab the actin and grab it like this. This move, see this move I'm doing? It's called the power stroke. Energy is needed here to push the myosin up, grab the actin down. This is the power stroke. See this? This is power stroke. And by the way, guys, last year in our exam, we got a question saying which of the following needs ATP? And there were some tricky answers, but the correct answer was power stroke. So that's why I mentioned it, even though you might not find it in this slide, but you'll find it in another slide, just written power strokes. Like, it's just very, we thought, oh, minutia, we don't need it, but no. Power stroke, I told you why, because we got last year in the exam. And this is the power stroke. See? This is the power stroke. Did I answer your question?
Yeah, but I'm still confused. Like power stroke is pulling the actin down, right?
No, it's not pulling. It's the movement. I will say it to you in a definition. It's the movement of myosin or the rotation of myosin forward towards the actin. This is the power stroke.
All right.
It's
So it's moving
towards the actin.
Tamam.
Because the actin is like this and myosin is like this. If it's granted from here, nothing will happen, right? We said that contracting is shortening of the muscle to pull the arm or any bone or muscle. So we need to go forward. This going forward towards the actin binding site is the power stroke, and we need ATP here. It's very important. You got that?
Yeah, thank you.
That's great, of course, ma'am.
Can you repeat it
Now
one last
here-
time?
Yeah, of course, I will repeat it. So as I told you, myosin and actin, you need to push the myosin towards the binding site so that the actin and myosin shorten. This pushing of the myosin towards the actin is called the power stroke.
Uh-huh.
Got that?
Okay, I got this.
Amazing, amazing. Look at this. This is also a cool thing. Now we said calcium left and is binding to actin and it's causing contractions and all of that. But we don't need calcium out all the time, right? This will cause unlimited contraction or maybe diseases, but you don't need to know the diseases now. So there's a nice mechanism we have that when we don't want to contract, don't want to use our muscles anymore, the calcium should go back to its storage. That cool? The calcium, we need to push it back. It's a very simple idea. We finished from the calcium. Thank you so much, calcium. Now go back to your home. Who does that? ATPase. ATPase just means that it highlights ATP. So it uses ATP to push calcium from a lower concentration gradient to a higher concentration gradient. Let me use some drawings here. Guys, you're gonna be bothered with my drawings, but I love drawings. The best way to explain is drawings. This is the sarcoplasmic reticulum calcium ATPase, SERCA. Of course, you heard of SERCA. This is it, SERCA. I think there's an E somewhere, but anyway, you know, sarcoplasmic reticulum calcium ATPase, SERCA. It pushes calcium. This is calcium. It's flooded outside, but inside it's even more concentrated. We need to push it all inside and concentrate it inside. صح? We said inside the SERCA, it's concentrated with calcium. We don't need any calcium outside. Now this ATPase will start pulling calcium inside. But there's also one thing I want to add to that, a protein. He's a helpful protein. He's trying to help. He's a nice guy. It's called calsequestrin. Don't laugh at me for the spelling. Calsequestrin, I think it goes something like this. Anyway, calsequestrin, look what it does. Look how cool calsequestrin is. Now, in general, what matters in the gradients is the free ions. So let's say we have 10 calcium here, free calcium, not bound to any proteins, free calcium. And we have 10 here, sorry, 10 Ca. Would anything happen, like equilibrium, would anything happen? No, right? Anyone disagrees with me? I don't think so. Nothing will happen. But say we have, which is in our case, we have like 1 to 10. And we still need to get this one inside. Okay, how? Calsequestrin does something really cool. It hides some calcium from inside. So this is calsequestrin. He's like this. He's an old dude. He's just trying to help. He takes in some calcium for himself. So this becomes free gamma 6. So the gradient is now lowered. It's buffered. This is the concept. It's buffered so that calcium from outside can, you know, go inside, not against a very, very high concentration gradient, just a bit lower. So calsequestrin plus SERCA, they help maintain a high concentration inside the sarcoplasmic reticulum. Is that clear? Is that clear, guys and girls, all of you? Here's calsequestrin, here's the SERCA. I talked about other things all written down there. I think that it's clear. Okay. Now, what I told you is the need of ATP. We said power stroke. It's written here. We said dissociation of myosin from actin, which means to basically just let go, right? When ATP binds to myosin, just lets go of actin. And for ATPase, SERCA, SRCA, ATPase, we need ATP for that. So these three things we need ATP for. I already mentioned them, so I should know them by now. Now, sources of ATP. Source of ATP, I think you know it, but I'll go over it anyway. So you have immediate source of ATP, which is basically free ATP. So if you have ATP around your room, you just take them, use them. The ATP finished from the room. You take out from your pocket some ATP. The pocket here is what? Where is it? Yes, creatine phosphate. It will get hydrolyzed, make more ATP. So when you go to the gym, at first you will use the free ATP, and then the free ATP will be depleted. You will use the ATP from the creatine phosphate. Okay, creatine phosphate ATP is depleted. Where do we get ATP from now? From, look at how cool this is. Let me go to the next slide and show you from where. Anaerobic glycolytic conversion. I think you took glycolysis by now. It's lecture 17 and more. I think you took it. Anaerobic glycolytic. You didn't? Okay, I got you. No problem. I got you. 1 second. I'll go again. Okay, look at this. So it's just glucose. I'll just like I will not go over the steps now. To pyruvate. This is glycolysis. And now this is step. See this step, lactic acid. And now you'll know why you get sore in the gym, because of lactic acid. High acid, you get sore in the gym. This process produces ATP, which is our third source of ATP. First, we said it was free ATP. Second, it was creatine phosphate. Third, it was, it is glycolytic conversion, anaerobic. So why it is anaerobic? What does anaerobic mean? Anaerobic means it does not use oxygen. So if it does not use oxygen, this is the pathway. Glucose, pyruvate to lactate. Don't know the pathway, just know that it's anaerobic glycolytic conversion. What is the aerobic glycolytic conversion? It uses ATP. So sorry, it uses oxygen to produce ATP. So pyruvate will go to acetyl-CoA. You don't know this. You're not supposed to. I'm just saying to make you understand. And this will go to the mitochondria, etc., etc., Krebs cycle, all of that, produce ATP. This is the fourth ATP source. So we said the first one is free, second is creatine phosphate, third one is anaerobic glycolytic conversion, fourth is aerobic glycolytic conversion. And inshallah, this makes it very clear, and it's really easy concept, this one. Yes, there is. Okay, now let's go to the next slide. If you have any questions, tell me, even if it's easy, it's fine. Oh, what's that? Okay. Okay, so we finished a good part of muscle physiology in 30 minutes, which I'm very impressed. Even we started late and you guys asked some questions. Anyone has any questions about what we just said? Anything? No
I have a question.
Yes, say.
So we said that the calsequestrin, it like holds calcium so that it's in the sarcoplasmic reticulum, right?
Yes, yeah.
Okay, so it just makes the gradient from the intracellular, like, sorry, what were you saying?
Yeah, look, it's just it's a buffer. You know what a buffer is? When you buffer
Yeah.
something? Yeah, the buffer. Calsequestrin is a buffer for calcium. So we have a lot of calcium inside the sarcoplasmic reticulum. It will hold some for itself to allow more calcium to come in and replace those. Basically, to take it in this way, that it buffers some to highly concentrate calcium, because we don't want any calcium outside, right? We don't want any outside. You got this?
Okay, thank you.
That's basically its job. It's a calcium buffer. This is 2 simple words. I hope that answers your question. Of course. Okay, so isometric, isotonic. I'm not sure how you guys deal with those. Are they easy? Are they hard? I'm not sure. But I hope you will get them now, like in a very good way. You will go teach after me. Okay, very simple concept. You have to say with me. You have isotonic contraction. Before we get to the fancy stuff, we need to just define iso and tonic. Iso, I think iso means 0 or like not changing or equal, constant. It means all of these things. Basically, there is no difference in iso between before and after. This is isotonic. And tonic is the tone or the tensity of the muscle. Tone of the muscle, if it's hard, if it's flexed, if it's not flexed. So isotonic, it means that the tone of the muscle is the same throughout the whole process of the contraction. So this is my phone. This is my phone. If I push it up like this, and then push it down, my muscle that I used to push it up was isotonic, which means the contraction of my muscle did not change throughout the process. I did not contract more or contract less. I contracted at the same level throughout the whole process of whatever I'm doing. So as you can see, in nerdy terms, I would like to say, velocity is more than 0 because I moved my phone. My muscle contraction is the same. And there's work, you know, if there's velocity, there's work happening. I move my phone up, this is physics. So basically just no contraction, no increase in contraction, but there is contraction, just the same contraction. I move my phone up and down. Now isometric, it's quite the opposite. It's increasing contraction, but there is no movement. So you go to the big guy in the gym and you find him deadlifting 500 pounds, like, I can lift that. And you go to lift it, oh, you can't. You're trying to contract, nothing. The bar is now moving, but your back muscles are trying their hardest. They're contracting, intensity is increasing. No movement. Sorry, big guy. Not bothering you again. You got this? Isotonic, isometric. I think you got this. So you can read no shortening, but increase in tension and no movement.
Can you repeat it once again, please?
Which one? Isometric or isotonic or both?
Both.
Okay, sure. Isotonic, the contraction of your muscle stays the same, but the object you're moving is actually moving, like there is movement. In isometric, the contraction is increasing, but there is no movement of the objects. Like this is a question actually we got last year. Last year we got, give us an example of, or what is an example of isometric contraction? And one of the choices had squeezing your glutes. I think you know what the glutes is. Do you know what the glutes is? It's just the back muscles below the hip. This is the glutes. So squeezing your glutes or squeezing any muscles, basically, you're not changing anything, but you're increasing tension as you go with time. You're increasing tension, but nothing's happening, you know? So you're increasing tension. So that's isometric. Isotonic, as I told you, just move something and it moves with you and there's no change in contraction. Yeah, I think that's all about isometric, isotonic. Okay, see, this is... You have
Just a minute.
question?
Yeah.
Yeah, sure.
Yeah. So in the constant tension contraction, you were raising your phone, right? But as you raise
Yes.
your phone, doesn't the contraction change of your muscle? It
Look,
gets
in
relaxed,
real
right?
life, yes, because gravity and all that, but what I'm trying to say here is that isotonic, it's just basically not changing the tension of your muscle, but you're changing the velocity of the phone. But yeah, in real life, you know, ideally the things will change. You're going to deep physics and telling me this distance. No, but don't go this far. Just understand the concept that you're not increasing your muscle tone or tensity or tension, but you're increasing the distance traveling by some object, whatever. You got this.
All right.
Thank you.
I have a question.
Yeah.
For isotonic contraction, is there shortening of the... Oh, never mind. It's written on the thing. Never mind. Thank you.
Yeah, no problem. It's fine. You guys got this? That's great.
Yeah.
Yeah.
Amazing. Guys, graph, we all hate it, but we get through it. It's easy, even though it might look, oh, what is passive tension, active tension graph going this? What is all this? I got you. Let me explain. So before we just get into all of this, let's set some ground rules here. So passive tension, actually before even this, now everything here is actually hypothetical or in lab. Like in your body, there is nothing such as passive tension. And you will know why when I tell you what is passive tension. Passive tension is basically, consider you have a muscle, just a muscle, casually lying on your hand, the biceps, and you fix it from an end and you pull it from one end. Now you pull the muscle, right? And the tension in the muscle increased because you're pulling it. See this pulling? This is passive tension because the muscle did not contract, but you moved it. So it's passive, it's not active. If the muscle contracted, the muscle itself contracted, that's active because the muscle is active. See? So passive tension, no one is gonna pull your muscle. Don't worry about this. This is all on lab. No one's gonna pull your muscle and oh, this is passive tension. You're probably gonna lose a muscle or lose a friend. Someone did it. Anyway, this is what this is all about. And there's something really cool after this, which will, inshallah, will understand. This is the same thing, but I want to tell you something here. See, I want to tell you something really important. I want you to focus, maximum focus. Look at this. Now, this graph has a lot of lines and stuff, you know, things we just look at, stare for 10 minutes and we don't know what's going on. It says compressed here. Again, this is a muscle. Fix it from one end, push it. It's compressed now. When it's compressed and you stimulate this muscle, the muscle will contract. But this, in theory, they say when you contract the muscle while it's compressed, the active tension is weaker than when it's in an optimal state. And it also says when the muscle is stretched, you stimulate it, and it contracts, the active tension is also less than the optimal state. And I'll explain to you why with a drawing. Let's show you a quick, nice drawing. Let's go down and I'll show you something really cool that you will get everything, inshallah, and hopefully. Let's draw some actin and myosin because you know muscle, it's all about actin and myosin. It's about 80% of it.
I'm really sorry for bothering you, but
It's fine, it's fine.
I don't really understand the passive and active.
Passive is basically the muscle itself is not doing anything. You're the one pulling it. It's passive.
Mhm.
Active, the muscle is doing some work. But as I told you, this is not in your body. In your body, as I told you, no one's gonna pull your muscle. Your muscle is gonna stay there for the end of your life. But in lab, go and must get a muscle, grab it just from a cadaver, put it on your hand, pull it. This is passive. But when the muscle itself does the work, it's active because the muscle is active.
Okay.
Let me show you just this cool, very cool. Why is this black? Say black for a second. Yes, okay. This is now myosin. 1, 2, and then 3. And 1, 2, 3. Guys, I'm not an artist, so this is something just I come up with. Here's the myosin is the red, and the actin is the black. Now I want to show you something really cool. This is the muscle in the optimal state. And if you ask me, what do you mean by the optimal state? I'll tell you what. The optimal state is basically it's the state of the muscle when its active force or its contraction is basically the strongest. So your muscle is the strongest in its optimal state. Okay, what is this? Don't worry. Look at this. We said when we stretch the muscle, it becomes weaker, and when we compress the muscle, it also becomes weaker, or its active tension in scientific terms, in active tension becomes less than the one in optimal state. Why is this? Look at this, look at this. Now we will compress it. See this thing? We will compress it, and I will show you what will happen when we compress it. We compress it. Black, bro, give me black. We compress it. It will become something like this. Almost the last one doing. Go here and 1, 2, and 3. 1, 2, and 3, and then this is the myosin. The red is the myosin, the black is actin. Notice something here. Notice something. Notice something very well. 1, 2, 3 contractions. But down, when we compress the muscle, what happened? Only 2 of the myosin. What is the mouse? Okay. Only 2 of the myosin are now binding. So we removed the existence of one myosin. This means it's one myosin weaker. And if we imagine this on every cell, this means the muscle actually got weaker when we compressed it. Oh, wow, that's nice. Huh? You guys got this? Why did it get weaker? And how the myosin just doesn't work now. It's just outside because it's very compressed. The myosin is now out and only few is grabbing onto the actin. Same thing happens when we stretch the muscle. when we stretch the muscle, let's stretch it. Let's stretch it out a little bit. Let's stretch it. Oh, that's very stretched out, you know. This is bad. Don't want to stretch out muscles, guys. I don't mean stretching, by the way. I mean stretching like in lab when you stretch the muscle. Look at this. This is 1, this is 2, and this is 3. Oh, this one is not doing anything. This is 1, this is 2, this is 3. Oh, again. This is stretched. Wow. You see that? You see this? This is binding perfectly. It's holding, like, put your fingers. Which one is strongest? This or this or this? See what's happening? It's strongest when you're holding with all the fingers. Likewise, the muscle. It's the strongest when all the myosin is binding to all the actin. But when you compress it, the myosin will not bind to all the actin. When you stretch it, the myosin will not contract or bind to the all the actin. Look, only 2. Only 2. These 2 are free. These 2 are free. Guys, please tell me you got this concept.
So is the optimum
Yeah.
length when it doesn't move at all, basically, when the muscle doesn't move?
No. Look, not move. There's a fine line between
Contract.
it getting, yeah. So there's a fine line between it getting compressed and contracted. When it contracts, it's contracted from the optimal state to the optimal state. But when it's compressed and it contracts, compressed, it's weaker. And as I told you, this whole thing does not happen in your body. Your body is perfectly fine. Everyone, alhamdulillah. This is in vitro, which means in lab. In lab, when they tested out the muscle, they found out that when you compress it and when they stretch it, it becomes weaker. That's in lab. In our bodies, it does not happen, guys. In our bodies, this does not happen. Your body is perfectly built, tendon to tendon, muscle to muscle. You know, it's perfectly built. This is only in labs. Like this, just scientists. You know, they found out something. They're playing around with muscles. They stretch it. They compress it. They found it's weaker. Why? This is because of this. Because the myosin, when it's compressed, is not fully bound to the actin. When it's stretched, the myosin is not fully actin.
So length does not get stretched when, for example, you're lifting weights or something, for example, in your arms.
No, this does not happen when lifting weight. No, not at all. When you're lifting weight, your muscle is always at the optimal state. As long as you're a normal human being, you know, and your muscle is not out of your body in the lab or something, you're completely fine.
Okay, thank you.
Yeah, this is only in lab. See, I think it says it down here. Changing the in situ muscle, which means it's just in lab. It's not physiological way the body regulates active tension. See, this is not the way our body regulates tension. Our body is always at the optimal range or normal working range. See, this is the working range. Now let's go back to the graph and apply what we learned from the drawing in the graph. Compressed. Where's the pen here? Compressed. Where's the pen? Here's the pen. Look at compressed. Look at the tension. Look at stretched. Look at the tension. Now look at resting or optimal state. Wow. Makes sense? Does that make sense, guys? Please be honest.
Yeah, it does.
Can you
I'm
just
actually proud of it.
repeat the compressed part when compression happens?
In the drawing or in the graph? This one?
In the drawing, please.
Yeah, sure.
Thank you.
Look, this is the muscle normally. Okay, this is the muscle normally. 1 second, just let me put down this. Yes, this is the muscle normally. When we compress it, this is what happens. See, we compressed it. See, these black lines, this is the actin. It got compressed. Now, not all the actin is binding to the myosin, which makes it weaker. Try it on your fingers. 4 fingers, strongest. Now, compress your hand. Boom, compressed. 3 is weaker. Compress more. Boom, 2, weaker. 1, weaker. And stretch it the opposite way. This is exactly how our muscles work. In lab, not when in our bodies, okay?
Okay, thank you.
Of course, and this is what's happening in this graph, as I told you. Now, about the total tension, it's nothing you calculate. It's just, I'm sorry, it's not nothing you measure. It's something you calculate.
Excuse me.
He just added. Say, yes.
يعني هو الconcept يعني yeah it's really easy بس يعني when you look at the questions it's very complicated. Can you
I'll
explain
come
that?
to you. I have actually some questions for that topic, especially that topic. I'll come to that and I'll show you how to deal with the questions. You'll see it's very easy. Trust me. Of course. So this is the normal working range, as we said. And the total tension, it just calculated the passive tension, because when you stretch it, you're increasing the passive tension. See, stretching, I'm increasing the passive tension. See that? So you add this passive tension to the active tension to the muscle, but I don't know why they put it there. I still don't know what purpose this whole thing serves, but it's part of your objectives, and I gotta explain it to you. And that's basically this. And see, now let's go back to our graph, our old friend. This is easy now, right? This is very easy. When we stretch it, passive tension increases, active tension decreases. Wow, very easy. This was supposed to be for drawing, but it's now for some reason it's lagging. Now this is exactly what I explained. I'll zoom. Oh yes, yeah, that's good. Look at the first one. It's compressed. If you notice here, I don't know if you can see it, there's 2 lines of actin. This is because it's compressed. So not all of the actin is binding to the myosin. Likewise here, but it's just less extreme in B. In C and D, you notice it's in the optimal state, but in C, it's a bit compressed. So the difference is not much. In E, it's stretched now. Look at the myosin heads. They're not all binding. See, they're not all binding. Look at the F. They're not even binding at all. At some point, there will be no tension as you stretch it out. No myosin touching the filament in this graph. So now let me ask you a question now. Let's put it down on the table. From A to E, which one is the optimal state? And why?
D. D.
C and D.
Wait, who said B? Who said E? And why? It's fine, guys. It's fine. It's fine. Just tell me why. Because I will tell you why not. Look at B. Before you say B, look at B. Do you think B, myosin, and action are all binding together? If yes, it's the answer. Do you think in E, the myosin and action are all binding together? If yes, it's the answer. But the answer is unfortunately no to both. But it's fine. Make mistakes now better than you make. Yes, the answer is D actually. D, because in the graph it's like more, this is a small extents, because when the larger image it will be like slight difference. But in the exam they won't get you C and D, they get you one of them. So they don't confuse you guys, because technically they're both right. Technically they're both right. They're both optimal state, they're both all binding together. Everything is working normally, nothing is going off or sloughing up and down. Everything is on the spectrum.
In B,
Sure.
in B, basically
Actin.
the actins are overlapping each
Yeah.
other. That's why they're not
That is
they're not in optimic. I'm sorry.
yes.
What I'm
because
trying to say,
overlap.
yeah. So like they're on top. The actin is on top of each other.
Yes.
And in,
So when 2 actins are on top of each other, this one is not working. So it's wasting muscle strength.
and the sarcomere length is short, right?
Shortness, it's not really the issue here. It's just about the myosin and actin overlapping. They all should be binding together. If something is not binding to the other thing, like if actin is not binding to myosin or the opposite, then something is wrong and the muscle gets weaker. We just showed it in the graph, and now here's answer D. We'll come to this graph again. I have a question for this exact graph, and I'm expecting all of you to get it right. You guys are mashallah so good.
Excuse me.
Yes.
Can you go to the first graph that you showed? First graph.
This?
Yeah. Can you explain the active tension in this, as it's just getting stretched, but still it's like decreased and then increased and again decreasing?
Yeah, because here we compress it. Now, as we stretch it, it becomes the optimal state, which is basically alone. It will just go back alone to the optimal state. In the muscle, if you don't touch it, it will be in the optimal state. Compress it, it goes to the left. Stretch it, which means increase the passive tension. Oh, increase the passive tension. It will go to the right.
Okay. Got it.
That's pretty much everything about this. This is, we covered this. And now this is like very easy. It's just basics. It says here Vmax, velocity and force. And as we see from the graph, decrease in force, increase in velocity. Increase in force, decrease in velocity. Let's carry this case. It's a robot's case. It's very light. You know, how fast can I contract? because the force on this one is very weak. I can contract it like very fast, Vmax, and F is very low. But when I go and, for example, grab my, let's say my bag is like pretty heavy, I can't do the same speed, right? Because the force I need to put is more, so the velocity will be less. That's the concept. So I'm not sure there's something significant here. It says it here in the text that lightweight is faster to lift, and high load or high weight, heavy weight, sorry, is just harder to lift and it will be slower. That's about the slide. There's not much here. This is important. What it's saying is that there is a time between the nerve when it is what do you say? When you want to contract, okay? The action potential traveling to the nerve, to the muscle, this has a little bit of time. So when you put an ECG and EKG, I'm not sure, I think you went to an EKG lab or something to measure the muscle. You saw that the nerve, you will see its action potential before you see the contraction of the muscle, because the nerve is faster and it happens before the contraction happens. This is the graph, but this is not where it's significant. Still, wait a minute. Tell you why this is significant, because of this. Look at this. What this is saying is, say there's two neurons and this is the muscle. One action potential, one action potential. As you can see, one, and there is one contraction. One contraction again. Okay, but that's not how we want it. Let's increase the nerves. Now it's 3 nerves. 1, 1, 1. But look, notice what's happened here. Start adding up the force, like this one. What did it do? It innervates my muscle, action potential. Now, followed by the second one, the force of contraction between the first and the second, they add up. Contraction is now stronger. The third one will even innervate and give an action potential, add up even more, until you eventually come up with this contractions. And this one, which happens in everyone, like how we contract, a lot of nerve fibers innervate the muscle, send action potentials, they all add up to a certain strength and stay constant, and still we relax. This is just the concept. And there is something here that they said is pathologic, which is clostridium tetani. I'm not sure if they like give you this stuff, clostridium tetani and gram-positive anaerobic bacteria. Do they give you this stuff? This bacteria and the pathology. I think it's too early for you guys, but anyway, this is just, it inhibits the inhibitor of the action potential. So action potential will not stop when you get this bacteria, God forbid. That's just a small clinical thing. I'm not sure why it's included in your slides, but yeah, that's basically it about this. There's nothing really great. It's just important to know that how contraction happens. Not one fiber, not two, not three. A lot of fibers innervating together add up contraction. Does anyone have a question?
Can you re-explain this slide one more time, please?
Yes. Okay.
I kind of don't understand.
Look, it's okay. So nerves, they go to the muscles. Now, when the nerve sends action potential or depolarizes or stimulus, whatever you want to call it, to the muscle, it causes contraction. Now, one contraction. This is not how we work. The way it works, a lot of nerves, like too many nerves, or motor endings, we can say, they go to our muscle, and they all send action potential together. So one contraction, it adds up to one big contraction, like this one in D. See, but the contraction is one big one. They add up. That's the concept. It's simple, yeah. Don't overthink it or anything. And it's called the trippy. If you want to, once its name is trip or trippy, whatever, tetany. That's clear?
I have a question.
Yeah.
Do all 4 processes, do they all occur in the body, or is it only D?
No, it's just showing you the also hypothetical electrical activity and how it happens when only 1, 2, 3. Normally you just keep contracting, and if you have no neurological problems, alhamdulillah, you will contract normally, and a lot of them will contract together to add up one contraction.
Okay, thank you.
Yeah. And this is what it's saying. if it's a single neuron innervating your muscles or muscle cells, there will be junctions between them. And this case in the smooth muscle, I think you know, but in the motor muscles or like skeletal muscles, there are a lot of motor recruitments, more than one nerve going to several muscles. You know, that's just what's saying. And here it's showing you the action potential and how this force is being summed up. A, B. Look at this big one. They get summed up. Boom. One big contraction.
Yani, all we need to know for this for these two slides is yani, the more force we want, the more units we recruit.
Yeah. Yes. That's
Okay.
one thing we take away from this one. Yes. And this is how things work. See, now you ask me what happens. One only, look at the contraction, because only one motor unit is working. If it's 2, if it's 3, if it's 4, it keeps adding up. And as you said, more motor units, more strength or tension. See, it's written down. Okay, let's move on. This is saying that, you know, we have muscles in your body and they have muscle cells. They also have motor neurons. There is a ratio between motor neurons and muscle cells. Now, logically, you would look at this graph and like, okay, what do I need from this? I'm not sure if you need anything from this, but just know that the eye muscles, you need a lot of motor nerves. Why? To be precise in where you look. Notice in your eyes, you can move it literally every 1 centimeter. You can move it in a very precise way. So is your hand. You move your hand in a very precise way when you draw, when you write. But in your tibialis anterior, you just stop walking. It's part of walking process with the tibialis anterior. So you don't really need to be that precise when you're walking. You're just walking straight, you move a little bit left, right. That's just the significance of having a lot of motor neurons compared to skeletal muscles. The higher the ratio of motor neurons to skeletal muscle, the higher the precision. Precision, yes. That's about the slide. Not much here. Any questions though? So any questions I can answer? No questions, that's great. Skeletal muscle types. I'm pretty sure when you look at like, oh, big graph, you're terrified, but no. I got you a very nice way that I won't even explain the whole thing, but you will understand why each one is the way it is, you know, like why twitch speed here is slow and then it got like time to drop max tension. Let me run you through a process. Okay. I want you to focus with me here. Forget the graph. Forget it. You will now understand all of it and be able to answer every question after this. Look at this. Let me first set some ground rules. You know, ground rules, we base on it. You have in your body types of muscles for a reason, for a good reason though. Slow twitch that are oxidative. What does oxidative mean first? Oxidative means, do you remember when I showed you the fourth what it was? Source of ATP? It was aerobic glycolytic conversion. So this is oxidative. It uses oxygen to utilize energy or to make energy in a simpler words. This one, you know, because using oxygen, it goes through Krebs cycle, etc., glycolysis, it goes a lot of things. So it takes time. So now if I ask you, if you're running really, really, really fast, will you use this? You would say no, you won't, because it takes time to use oxygen. Plus, when you're running, your oxygen depletion increases, which means your oxygen in your body just decreases. So you don't use this. Where do you use this muscle? Where do you use this muscle that is slow, and uses oxygen for energy? And when I say oxygen, it's a long process. You use it for when you're standing. You can just stand up all day and stay there for like 5 hours and you're good. Because this slow twitch, using oxygen all the time, you're breathing in, they're using the oxygen, you don't feel sore. This soreness means acid has been released. So when you use oxygen, you don't feel sore. This is for the slow twitch muscles. Now there's fast twitch, but oxidative. Like they're fast, but they still use oxygen. This is when you're walking, you know, you can walk for a pretty long distance, you know, you can go like 2,000 steps without getting sore or anything, you're fine. Why? Because you're using oxygen and you're walking using fast twitch muscles, but oxidative. Where do you get sore? You know what sore is? It's when you feel a burning pain in your muscles. In the fast twitch, that are glycolytic. What does glycolytic means? It is the anaerobic conversion, which means the glucose will become converted to lactic acid for ATP, not oxygen. There is no oxygen here, lactic acid. And when we say no oxygen, this means no mitochondria or less mitochondria, which is why, by the way, guys, chicken legs are red and chicken breast is white, because chicken legs, they stand all day, they need oxygen, they're slow twitch, they're red. They need mitochondria, oxygen. Chicken breast, it's white because they don't use chest all day. How many times a day do you use your chest? Not much, right? So it's fast twitch and white, glycolytic. So when you go to the gym, bench press, start 1, 2, 3, 4, start feel sore because your muscles are using fast twitch glycolytic fibers. They're utilizing glucose to lactic acid, which produces more acid, make you feel sore. This burning ache, we all hate it. Also, when you're sprinting really fast, like your friend just grabbed your phone and went away, you just want to grab him and take his phone. Just don't be violent, okay, guys? I'm not supporting violence. I'm just saying you will be very fast. This sudden burst of activity, as you can see down the graph, sudden burst of activity being very fast, you're using fast twitch and glycolytic because you want to utilize the energy fast. You're not waiting for oxygen. I'm not here to wait for oxygen. Use it very fast and try to get up fast and just sprint all you can. You will feel sore immediately. So now let's look at the graph. Now, leave me alone and focus on the graph. Look, stay with me, guys. Stay with me. Twitch speed. In the slow oxidative muscle, do you think twitch speed will be high or low? It will be slow. What about the time to derive maximum tension? You're standing. How long will it take to reach maximum tension? It doesn't reach maximum tension. If it does, it takes a lot of time. So it's slow. What about contraction duration? How long can it stay contracted? For a long time. You're standing, it won't stop. It's a very long time. What about myosin ATPase activity? Now, myosin ATPase, it's the one we said for the power stroke to hydrolyze the ATP to utilize the contraction of muscle. So, do you think this would be fast or slow? And I would say slow because it takes time to contract. It doesn't contract immediately. Don't need it to be fast. Slow. What about the SR content? Will be high or low? You want high calcium or just low calcium? And for me, I would say low calcium because you're going slow. So it's slow, You don't need to be very fast. You don't need to have a lot of calcium. You don't need to have high myosin ATPase activity or anything. What about the glycolytic capacity? It's not using glycolysis. It's using more ATC and Krebs cycle. Mitochondria, because it's using ATC, Krebs cycle, it's using oxygen, high mitochondria, which is why chicken legs are red. So you keep that in mind. Myoglobin, same thing. Myoglobin transfers oxygen. Capillary, you need oxygen, so to be dense. Fiber diameter be small because you're not contracting for strength, you're not contracting for speed, you're just contracting for stability. Fatigue resistance very high. You can resist fatigue. You can, stand all day and you're not fatigued, you're not tired, you're not sore, you're still fine. So it's very high. Now look at the fast twitch. Just look at it. I'm not going to go all over again. Just look at it and tell me if it makes sense. Just take a look. And I think it makes sense because it's literally what you want when you want to sprint. Go fast as you can.
Do we
Does
have
anyone have
all of these?
You don't have to memorize. Don't memorize. Understand it. Okay, I will not show you the graph. For a fast twitch muscle, fast twitch, which means you want to get up fast, you're using glycolytic energy source. Do you think the mitochondria will be high or low?
Low.
There you go, guys. Yeah, I told you guys are amazing. It's slow. Why is it low?
Because it's
the
using
fast
glycolysis.
Because you're using glycolysis, you're not using oxygen. Remember, guys, mitochondria, oxygen. Mitochondria, oxygen. Because mitochondria uses oxygen for energy. So if you're not using oxygen, you don't need much mitochondria. Just like the white chicken breast that you enjoy at your home, at your dinner, whatever. Okay, let's ask you another question. Let me ask you, myosin ATPase activity in the fast twitch muscle. Do you think it will be fast or slow? Basically, the contraction.
Fast.
See, you don't have to memorize it. You just think about it. What does this need? You can test yourself, and Just think, okay, fast switch, what does it need to have? This, that, this, that, this, that. You look, oh yeah, you got it right. MashaAllah, you guys are amazing. And this is the last slide. After this, we're going to do some questions. Oh, I spoiled, sorry. Some questions. Okay, this is question. This is I got from Ambus. For those who are familiar with Ambus, I think you guys know Ambus. This question is from there. And it's a pretty good question. I think you guys should get it. I'll give you 30 seconds to read. You should get it in 30 seconds or even less.
Is it D?
Why?
Because detaching the myosin head from actin requires energy.
Straight up recall. That's amazing. Good job. And that's right. It's D. If someone is confused or is not convinced in the answer, you can say, you can open your mic and we can discuss the question. I don't mind. I think that says it all. You know, you guys are amazing. Let's go to the next question. Can you guys see the question? It's pretty far up, you know. You guys can see the question?
Yeah, we could see.
Okay. Okay. If you got the question, you can tell me the answer.
Is it A?
And why is that?
Because
the
it's storing
calcium- Okay.
because storing calcium inside the sarcoplasmic reticulum causes the muscle to relax because calcium activates muscle contraction.
Perfect. And by the way, this was from Guyton test bank. So Guyton, it's just known for not so easy questions. Like this question is decent. It's decent. It's easy for you guys because you now you understand, you know what's actually going on. Let's go to the next question. And this is the wait one
I have a
second.
quick question.
Say.
Why
Say.
can it be C?
C, what is C? Depletion of calcium stores from sarcoplasmic reticulum. When you contract and relax, do calcium stores in sarcoplasmic reticulum get depleted? And if we deplete the calcium stores, will that cause relaxation? It will cause a pathophysiological problem, which is another thing, but precisely it should be A. You know why it's A? Are you still here? It's fine. It's fine. We're learning all together. It's fine. You know how many mistakes I did? You can't count them. So you can just discuss this question with me. It's fine. I'll accept your argument. Okay, I think you understand then. Okay, we can
no,
move.
actually, no. She asked in the chat like, why is it A?
I can't see the chat, guys. I'm sorry. I'm really sorry. Why A? Okay, I'll tell you why A. Now I'll open the chat. Okay, now it's better. Why A? Okay. As we said, when we want to relax, two things that we do to relax. First, we bind ATP to myosin so that myosin gets back down. The second thing, we activate the SERCA, which is sarcoplasmic reticulum ATPase, calcium ATPase, to grab the calcium from the sarcoplasm to the reticulum. By doing that, it decreases the sarcoplasmic calcium so that the actin does not get activated by the calcium. So relaxation. Now you got it?
Yeah, thank you.
Amazing, thank you. Of course. Okay, yeah, it's A. I put it there, and now 24. I'll give you some time to read this question. Take your time on this one, because it's needing you to think. If you know the answer, just wait a bit for your students, for your batchmates, sorry. Did anyone get it?
It's increase, decrease.
Okay, wait. Did you get it? Just say yes or no.
Yes.
Yeah.
Yeah.
Okay. One of you answer. You guys agree on someone and answer this question. Okay, one tell me about the passive tension. Someone tell me, Hassan, you said something. Continue. Passive
Yes,
tension.
I
Why?
said,
Why passive tension?
Passive tension increases because the longer the length, the longer the stretch.
Nice. Now someone else tell me why the maximal active tension will increase, decrease, or not change. Tell me.
Decrease because
Sorry,
it's
say it again. I didn't think. Hey,
decrease.
decrease. Why is that?
Is it because of the overlapping of the? It's overlapped by 6 centimeters to facilitate the suturing, which means that the myosin and actin are all attached to each other.
You understand it, but the way to say it is just
Can
because
I
it's stretched. The lens
Can I
is more,
explain?
it's more stretched. When it's more
Oh.
stretched, as we agreed, what happened to maximal active tension?
Decreases.
Yes, salam. That's amazing. That's amazing. Anyone does not get why it's D?
I
I have a question.
Yeah, ask.
So for overlap, isn't that like, I don't know, when I thought overlap, I thought compression, like it was shortening.
No, what I said is this tendon, it got cut. So
Yeah.
they put a tendon from its behind and stretch it to attach it to the attachment site. Because what happened? They attached the tendon from the back, so they stretched the muscle.
Oh,
When
so
they
the
stretch the
length is
muscle-
increasing, so the passive
Exactly.
tension is decreasing.
They
Okay,
go- no,
sorry,
passive tension.
yeah,
Wait,
passive tension is increasing, yeah. Okay,
that's right. You got this
thank
right.
you.
MashaAllah, you guys are doing very good. That's a very good sign. And I'm hoping you all get A's in the exam. Inshallah, I get all A's. Let's go to the next question. We're not done yet.
Please repeat this question?
Yeah, of course. So, man had an injury here in this tendon, okay? This tendon. Now, what the doctors did, they decided to, the tendon, it's a long, like musculofibrous structure. So they can just take part from the back and insert it forward. Let me draw. Actually, let me draw. Look at this. This is the tendon normally. Normal cases, this is the tendon. This got cut. See the line again. Now, what they did is they got this part and they stretched it out to the attachment site. So now this one is stretched out. And when we said it's stretched out, what happens to passive tension?
Increases.
We stretched it. What happens with active tension when we stretch it?
Decreases.
Decreases.
You got it. You got it. Let's this. You got it. MashaAllah. Good job. Good job.
Just a question.
Of course. Say.
Can we think of passive tension and active tension as inverse proportion? Yani when one increases, the other decreases.
Yes. Look, but it's if you decrease passive tension from a point to a point, because it's relative to the points. If it's stretched and you decrease passive tension, what you're doing is increasing active tension. But when you decrease it even more, it decreases. So not always. Just stick to the idea that if it's shorter or longer, and then think about the terminology. I don't like you to only memorize. I don't want you to memorize in physiology. I mean, you should understand. So so don't do that.
I
Maybe
get
you
the
get
basic
confused.
idea of this question.
Yeah. Look,
like if
to some extent, yes, you can. In this question, yes, you can. But just be careful from the tricky questions. I'm just saying this because if you get a tricky question, you might play with words, and if you play with words, it might trick you if you only memorize the words. If you understand, it will never trick you.
طب معلش
You got
بس can you give a quick recap of passive and active, because I get the
Yeah,
basic
of course.
concept, but in this question,
Of course.
just to get it.
Of course. Don't worry about this. Do you see my wait, do you see the thing?
Yes.
Okay. We said that this is myosin. Now, active tension is when you stimulate the muscle itself and it contracts. This is active. See, the muscle is active, it contracted. When you pull the muscle, who's doing the work? You or the muscle? You pulled the muscle. Who did it? You or the muscle? You or me. You, yeah. So that's passive because the muscle didn't do anything. It literally just didn't do anything and you pulled it. That's passive, right?
Yeah, I get it.
And this is just saying that if it's compressed, stretch, same idea. You got that?
Yeah, thank you.
100%?
Yes, yeah, yani, but basically like passive yani, the muscles just stressed, and active, the muscles yani is stressing itself, right?
That's 100%.
Okay.
Yeah, the muscle doesn't do anything. You do the work, not the muscle. I mean, you do the muscle, not your muscles. Careful.
Okay.
Okay, let's go to the next
Thank you.
question. It's of course, man, of course. This is a cool question, and I told you I'm gonna get you a question, same as the graph that we saw. You should solve that 100%.
Is it C?
Give it 10 seconds. Let's just 10
Yeah,
seconds for your batchmates to get the answer.
It's safe.
Okay.
You guys all got it?
Yeah.
That's less than 10 seconds, mashallah. You answered this question in like 3 seconds. And you're right. It's absolutely right. It's C. We agreed. Optimal length, optimal strength, maximum strength, whatever you want, is at the resting, which is the maximum in the graph. I think that means that you understand everything.
I just have one. I just want to say something.
Say.
So E is basically the start of the passive tension.
E is when you stretch it out to the max. You stretch it out until there is no myosin in contact with the actin. See, you stretch, they're not in contact.
Okay, thank you.
Of course, man.
In passive tension, I just want to ask you, in passive tension, what was I gonna say? Yeah, in passive tension, so like there is no length, so like the tension is greater and the length of the sarcomere is smaller. So we can, for an example, lift things with short length, right?
I think you're mixing things up. Passive tension doesn't need all that. Passive tension is just the tension that you exert on the muscle. Just think about the pulling of the muscle. Passive pulling of the muscle. That's it. That's it. Very, very simple. Don't go too far. That's basics.
Thank you.
Of course, man. And this is the end of it. If you guys have any questions, anytime, you're welcome. I'm happy to help you. And if you have any questions now for anything in the lecture, I'm also happy to help you. If you're done and you want to leave, you can leave. I hope you, inshallah, the best of luck in your MSK midterm and final, inshallah. And yeah, that's it for today.
Thank you so much.
Thank you so much.
Of course, you're welcome, guys. So any questions? No questions? All right, then I think that's it.
Oh, wait, I have a question.
Say.
The last one.
Yeah.
8. I don't actually get it. I know it's
that's
easy.
fine. Yeah.
Right.
You remember this? You remember this one, right? 1 second. You remember this graph? Can you appreciate the relationship between the actin myosin overlapping and the graph, which is basically saying the higher you go with the tension, passive tension, or the stretching, the lower you get with the maximum tension, the active tension. And the opposite is true. When you compress it, what happens? Decreases. So what is the optimal length or the maximum tension? Where is it achieved? You tell me, where is it achieved?
It's at the optimal length.
Which one in the graph? Which letter in the graph?
The one that has maximum tension.
Say the letter. It's A, B, C, D, E. You can see them, right?
C and D.
You got it. You got it. Now we will go back to this question. Which one's the
Okay.
optimal? Yeah, C. It's very simple. Yeah, of course, man.
All right.
Any more questions? Of course. Thank you so much, guys. Thank you, MSA, the GND, Karama. Thank you. I think now we can leave, right?